Hello, today I'm going to talk to you about amino acids and proteins. There's going to be huge emphasis on protein and understanding protein structure and function. So let's get started. Now, when we talk about the idea of proteins, we're transitioning to a world that features biological compounds and the use of chemistry as it pertains to organic chemistry or functional groups. The branch where these two intersect is biochemistry because it is the study of molecules and their reactions in living organisms. Many times, physicians are faced with biochemistry decisions every day. Any disease are associated with abnormalities in biochemistry, which is especially pertinent in the world today. Now, nutritionists evaluate dietary needs based on the biochemistry of the body. The pharmaceutical industry designs molecules that can mimic or perhaps alter the action of biomolecules found within biological systems. The goal of biochemistry is to better understand the structures of these biomolecules and the relationships between their structures as well as their functions. Now, like I said, it's the common ground between life science and chemistry. Now, the principal class of biomolecules that get discussed generally are proteins, carbohydrates, lipids, and nucleic acids. This presentation is going to focus primarily on proteins. Now, the biochemical reactions must continuously break down food molecules, generate and store energy, build up new biomolecules, and then excrete the waste. And despite the huge size and complexity of some biomolecules, their functional groups and chemical reactions are really no different than simple organic molecules. And so all of the principles of chemistry that have been discussed basically apply to the world of biochemistry. So that's the reason for our introduction. So it starts with kind of housing some familiarity with some of these functional groups that we may or may not have seen already. The first group is called the amino group. We've talked about it, and it talks about the amine group, where you have your nitrogen and it's fused to your hydrogen groups. You then have what's called a hydroxyl group, which features a capital O and H. You next have a very popular group called the carbonyl group, which is going to be found all throughout the discussion of biomolecules. You also have your carboxyl group, which will be very vital when we start talking about amino acids and proteins. You have your amide group, which defines a specific type of connection between the carbonyl group and a nitrogen, and we'll talk about that in more detail. You have some auxiliary extra groups as well, which include your carboxylic acid, your phosphates, your hemacetals, and your acetyl groups. And so let's go ahead and get started, and we want to really talk about that function and structure of a protein. Now, we've heard the discussion of a term protein, but I want you to understand that protein constitutes basically an amalgamation or accumulation of a bunch of amino acids. They form this long polymer chain. And we'll talk more a little bit about how polymers form of proteins a little bit later, but just understand that the precursor to proteins are amino acids. Now, every amino acid has two main groups that you need to be aware of. One is called the amine group. The other is called the carboxyl group. I've referred to it sometimes as carboxylic acid group, which is very synonymous with the same thing. Now, in addition to this, the amino acid groups always have some type of R group, which is called a side chain. Now, the focal point of where the reactions happen are at the amine and the carboxyl group, but what gives the amino acid its identity is going to be the side chain. Now, the central atom, which in this case is a carbon, is called the alpha carbon. And that's the carbon that's usually directly next to your carbonyl group if you're looking at your amino acid. Now, amino acids and proteins are alpha amino acids because the amine group H is connected to an alpha carbon. So each amino acid has a different R group, which I've already highlighted, which is found on the side chain. That gives it its unique identity, its property, its reactivity, which we'll go into a little bit later. R groups can be hydrocarbons, or they may contain a specific type of functional group. Now, when you put two or more amino acids together, it's called a dipeptide. D-I-P-E-P-T-I-D-E. Now, in order to form these dipeptides, they have to go through a reaction where the carboxylic acid, or the carboxyl group, reacts with the amino group or amine group to form a peptide bond. Now, interestingly enough, we've been introduced to this functional group as an amide, A-M-I-D-E. Now, with this reaction, the main focal points you need to understand is that the carboxylic acid group reacts with the amine group of another compound, and this reaction should be vaguely familiar. Now, this reaction is very vital as it relates to that of the reaction of peptides, which later come together to form your large polymeric groups, which are proteins. But before all that happens, it just starts with two independent biomolecules reacting together to form a dipeptide. Now, what you also should take from this reaction is that if you look at the reaction, the product formed is water. So what this reaction should tell you is that this reaction undergoes a process where water is removed. Now, as I highlighted prior, two amino acids come together to form a dipeptide. When you put three amino acids together, it is called a tripeptide. Now, when you have several amino acids that are connected together, they can be called a polypeptide. And there are other terms for peptide. There's one called an oligopeptide, which means like several. Let's say you have like 7 or 8 of them. But the point is, once you get beyond 2 and 3, they're considered polypeptides. Poly meaning many. Now, there are 4 levels to the structure of a protein. Because remember, we've talked about the amino acid as being kind of the precursor to a protein. Now, in the stages of protein formation, you need to understand the 4 various stages. So, the 4 are primary structure, secondary structure, tertiary structure, and quaternary structure. And I'm going to talk about each one, how each one is unique, and how each one plays a vital role in the overall formation of a protein. Now, the primary structure just represents the general sequence of amino acids in a protein chain. Basically, if you have a bunch of molecules that are amino acids, they all connect in a certain kind of series. And each one has their own characteristic functionality, speciality. You could have two peptides, you could have 2 amino acids that come together to form a structure, and depending on the order that they're listed, changes the whole integrity of the structure to begin with. So you have your primary structure, then you have what's called your secondary structure, which is kind of a regular repeating spatial organization of your neighboring segments in your single chain. Then you have your tertiary structure, in which the shape of the protein starts to produce regions that are bending and folding, so to speak. And then you finally have your quaternary structure, which refers to the overall structure of your protein, where you have kind of a combination of more than one polypeptide kind of fused together. So that's kind of a general synopsis. We are going to reapproach the subject, but before we do, one of the things we want to highlight is the various functions of different proteins, because maybe you've heard the term protein and not been able to understand it in context of its practical application or natural occurrence in everyday living. So there are several examples, and I'll mention the functions as well. So one of the types is called the enzymes. And if you've ever heard of enzymes, they catalyze or they speed up, if you want to use the word speed up, biochemical reactions. Now, they facilitate this through a number of ways. They lower the activation. They make it so that it's available for the reaction to occur. There are a number of things that happen, but enzymes are found all throughout our body, biologically. In fact, one of them is responsible for the digestion of carbohydrates, and so it helps break down food. Another type of protein is hormones, and basically, I'm sure a lot of us are familiar with that. They can be used to regulate body function by carrying messages to various receptors in the body. And one such example is insulin, although there are many, many, many others. Another one is called storage proteins. They make essential substances available when needed. And so for one of them is myoglobin. It tends to store oxygen in our muscles. You have examples of transport proteins, which carry substances through body fluids. You have structural proteins, which provide mechanical shape and support. Collagen, which is helpful in the body's muscle formation, etc. You have protective proteins, which defend the body against foreign matter. Immunoglobulin, we sometimes call it IgG. Antibodies, if you've ever heard of antibodies, that's what comes to mind when you talk about protective proteins. And then you have contractile proteins, which do mechanical work. You have examples of myosin and actin, which tend to govern muscle movement. So as you can see, there are various types of proteins, and the general term for protein is not a one-size-fits-all glove, but carries a myriad of things and functions that it can execute. So let's go back to these amino acid precursors before we start talking about really detailed focus of understanding how they come together to form proteins. So in all, you got about 20 amino acids. And each amino acid has a 3-letter shorthand code designation. Now that's kind of an abbreviation for what the real word or name of that particular compound is. Now for 19 of the amino acids, only the identity of side chain attached to the carbon differs. So that means that each one will have a specific amino group, each one will have a carboxylic acid group, and then that side chain that I mentioned earlier, which is what makes it unique. Now the remaining amino acids out of the 20, which is just that one, has a secondary amine whose nitrogen and carbon atoms are joined in a 5-membered ring. So it's kind of unique functionally when it comes down to the amino acid group. Now fortunately, the focus of this presentation is not for you to become overindulged in memorizing the amino acids, but just understanding the general function of how they interact with one another to form compounds or proteins, if you will. So let's look at them generally. You have a group, and this group is classified as your non-polar neutral side chains. Now what that means when they're talking about non-polar is that it doesn't have kind of an overt positive or overt negatively charged compound. And so when these amino acids come together, they tend to influence the behavior of the biomolecule they are forming. So what these compounds are saying is that they are neutral side chains and they will not influence them from a pH standpoint where they'll make it negative or overly positive. And the examples included here are alanine, glycine, isoleucine, leucine, methionine, phenylalanine, proline, tryptophan, and valine. And while I won't get in the habit of mentioning all of them, I want to kind of take focus on the functional groups. Now, each of these has the same amino group, which is in blue, and the same carboxyl group, which is in pink. But what makes them unique each is the side chain, which was mentioned before. If you look at the side chain of each of these compounds, which are highlighted in green, you'll notice these are what actually makes each one unique to another. And this also influences whether the compound is regarded as nonpolar or neutral, if you will. And so many of these compounds feature just a bunch of carbons in arrangement. And when we're saying nonpolar, it means that they're not causing specific interactions with other compounds in copious amounts. If anything, they will contribute to creating what's called hydrophobic regions, which we are going to talk about a little bit later as well. but these are your list of nonpolar neutral compounds. So just kind of, if I were you, I was creating a mini list of my amino acids. This would be part of my nonpolar compounds. Then you have your polar compounds. And these polar compounds create a number of interactions. Some of them might be hydrogen bonding. You have some thiol bisulfide bridges that can occur. There are a number of reactions that can happen, which we'll talk about a little bit later. But notice, again, with these particular amino acids, you have the amine group, which is pretty much the same in all of them, blue. And then you have your carboxyl group, which is pink on the right for all of them. And what makes each one unique is that they have the green color to indicate their different side chains. Now, one of the things that I want you to take note of is how many of these guys feature the functional group oxygen. And when you think about it, oxygen has kind of a habit of contributing to a compound having or exhibiting polarity. And so when we're talking about polar compounds, a good rule of thumb is looking for these compounds that feature side chains that have oxygen groups present. Now, uniquely, if you look at the cysteine, it is the one group in this section that does not have an oxygen group with it. Well, if you're somewhat familiar with your periodic table, sulfur and oxygen are part of the same family. And that means they exhibit similar reactivity. And so cysteines tend to interact with one another to form these things called disulfide bonds. And we'll get into that in a little bit more detail. But you do want to understand that they, among the other compounds, also exhibit a habit of being polar and tend to have polar interactions. So this list includes those compounds. Moving on, you have your acidic side chains. And with the acidic side chains, they tend to exhibit a little bit more of a impact on and of an acidic nature. And it's a very small group, interestingly enough, conveniently enough, if you will. Their names tend to have the word acid present, which makes it a little bit convenient. You have aspartic acid and glutamic acid. And notice that each of these side chains features a carboxyl group. So it's almost like having an amino group that has two carboxyl groups present. Now, when you look at the basic compounds, the group in the basic compounds tend to feature these nitrogen-containing compounds. So in your acidic compounds, they tend to have the carboxyl, and in your basic compounds, they tend to have the amine groups. Now, the 20 amino acids that make up proteins are either classified as neutral, acidic, or basic, which we've highlighted by looking at the images. The 15 neutral amino acids are further divided into those with nonpolar side chains and those with polar functional groups, such as amide or hydroxyl groups in their side chains. Now, the sequence of amino acids in a protein and the chemical nature of their side chains is what helps direct their functionality. And that's kind of what I highlighted when I was talking about those side chains. The side chain, or the green, is going to dictate kind of their interactions and their behavior as they begin to form these polypeptides and later what we call proteins. Now, non-covalent forces tend to act between different molecules or between different parts of the same large molecule. What that means is that they form interactions but not formalized bonds. And that's going to contribute to the overall behavior and identity of the amino acids when they form proteins. Now, nonpolar side chains are described as what we call hydrophobic, which is water-fearing. They tend to avoid aqueous fluids, and they tend to kind of gather or cluster together to create an environment that is water-free. Conversely, you have something called hydrophilic, which means water-loving. And those represent your polar, acidic, and basic side chains that tend to be attracted to water molecules. And they fold outwardly to be a part of water because they tend to be highly soluble in water and help with water solubility for overall protein function. So Let's look at the primary structure of a protein. And the basic building block of a protein starts with just the amino acids. Now, along what's called the backbone, you just have a straight chain of alternating amino acids. It starts where you have your amino group on the left and you have your carboxyl group on the right. And they always form in that kind of sequence where you have your amino group on the left, and we sometimes call that our N-terminal, and then you have the carboxyl group on the right, which is usually called your C-terminal. So whenever you're looking at the protein, its primary structure just represents the sequence or the order of those amino acids. So if you decided to put a bunch of amino acids together in a sequence, that forms the primary structure. Now, interestingly enough, when you look at just the primary structure, you can have 2 or 3 amino acids, and they form various types of proteins, because it's all about the order and not always just about the specific amino acids coming together. The way that I relate this is how we use words in the alphabet. You can take a word that has 4 letters, and you can use those same 4 letters to create a new word. Well, because you created a new word, that new word has a new function. And so when we're talking about the analogy here to amino acids, we're saying that if you put an amino acid in a different sequence, you form a completely different peptide compound. Now, as I mentioned, the carbon and nitrogen atoms along the backbone lie in a zigzag arrangement because they're kind of interacting, you're reacting them together. The carbonyl group, double bond electrons are shared with the adjacent carbon-nitrogen bond. This makes the CN bond sufficiently like a double bond that there is no rotation around it. So what this is basically telling you is that when you form a dipeptide, you form an amide bond that remains very structurally sound. It doesn't rotate, it doesn't wiggle, it doesn't move. And when you want to think about it, you want to think about the relationship between how hard it is to rotate going from a double bond to a triple bond. If you have a single bond, usually the bonds can rotate really easily. If you have a double bond, you're going to have a little bit more of trouble. And then when you move to a triple bond, you're going to have extensively more trouble trying to rotate that because it will have hindered movement. And the same thing can be seen when you're talking about an amide bond. Now, the carbonyl group, the amino group, or amine group, bonded to it, and the 2 adjacent alpha carbons tend to form a rigid planar unit. Now, what does that mean when we're talking planar? We're saying that if you took a sheet of paper, it would be all in the same plane. It's all kind of lined up. So, one of the things I want to reiterate, which I kind of said earlier when I was talking about it, is that when you put those 2 amino acids together, you can form 2 different dipeptides. And so if somebody gave you 2 peptides, X and Y, and they asked you how many primary structures can one form, you can actually form 2 primary protein structures, because you can have X and Y, or you can have YX. Now, obviously, this becomes more complex when you start talking about using 3 or more peptides, because it's almost like, you know, how many different combinations of a series of numbers can be ordered. I used to use, like, lottery numbers. If you use 369 and you use those numbers, how many various combinations of 369 actually exist? Well, there's a lot. It's not just 3, it's not just 6. It's quite a bit, because you can start with 369, 396, then you can migrate and flip it and say 639, 693, and you can flip the order and keep going. So you can create a large number of those various peptide sequences. Now, peptides and proteins are always written with the amino terminal, which is called the N-terminal, like I said before, on the left side, and then you have your C-terminal on the right. Now, the individual amino acids joined in the chain are called residues. So when you look at just the individual amino acid, it is called a residue, and the residue comes together to form a dipeptide. Now, a peptide is named by citing the amino acid residues in order starting from the N terminus on the left and ending at the C terminus on the right. Now, the primary structure of a protein is the result of an amino acid being lined in precisely the correct order. So it's crucial for primary structure to function that the change of only one amino acid it's key to understand that if you even change one of the amino acids, it can drastically impact performance of a protein or its biological functions. In fact, one such example, you're talking about sickle cell anemia, is the result of a single amino acid substitution that replaces one amino acid. In this case, glutamate with another valine in the hemoglobin molecule. So the amino acids are very important, and even one mild change can have a drastic impact. Now, a hydrophobic pocket is exposed on the surface of hemoglobin, and hydrophobic valine on another hemoglobin molecule is drawn into its pocket. So the one single modification changes the behavior of the overall functional group of the protein and changes it in a way that allows for another molecule to become a part of it as opposed to the original. And so this impacts the insoluble fiber chains which are formed, and then it forces the cell into kind of this sickle shape. And so it's a classic example of the impact of even changing one amino acid sequence. Now, as we know this, proteins are a necessary part of daily diet because our bodies do not store proteins like they do carbohydrates and fat. And 9 of the 20 amino acids must be obtained in the diet. And these are known as our essential amino acids. A complete protein source provides each of our 9 amino acids, and most meat and dairy products tend to meet these requirements. If you engage in vegetarian food diets, usually they supplement this with some other resource to get the energy they need. Now, there are disorders that can arise from inadequate protein intake, and that one of the terms is called protein energy malnutrition, called PEM. When you have certain cases like this, there's a number of them where they can either cause effects of the liver or other impacts. So one of the goals is always to make sure that our protein intake is adequate and sufficient. So let's talk about the interactions that help influence the shape of proteins. One of those things that I mentioned vaguely was that of hydrogen bonds. They represent non-covalent, where they're not formal bonds of atoms, and these hydrogen atoms interact with amino groups and the oxygen atoms in carboxyl groups, or carbonyl groups. Now, hydrogen bonds of our groups with each other or with backbone atoms tend to interact. Now, some amino acid side chains contain atoms that can actually form hydrogen bonds, and they can connect different parts of a protein molecule, and this can contribute to the structural integrity of a protein. Now, often hydrogen bonding side chains are present on the surface of a folded protein, where they can form hydrogen bonds with surrounding water molecules. So let's look at the shape and how they can be influenced, because this part is very important. If we're looking at determining the shape and the impact of the amino acid sequence, if you're starting from the left, you notice the pink dots, and these highlight your hydrogen bonds. Now, hydrogen bonds are not formal bonds. That means they kind of interact. They're going steady, but they're not committed. There's an interaction with them, and they only happen when you have a hydrogen group and the presence of a nitrogen or an oxygen group for when we're talking about amino acids. Now, these types of hydrogen bond groups can definitely happen when you talk about the hydrogen found in an amino group and the oxygen on the carbonyl group, as highlighted by the arrow over here. And it happens more often than not because you have several locations where this interaction can occur. Now, another type of non-covalent interaction that can occur is hydrophobic interactions. And hydrophobic interactions are deep pockets where they're like, we do not want the presence, or we would prefer the presence of water be excluded from us. And so when you talk about these hydrophobic interactions, there's a list of those compounds that fit the mold. And when you get a group of them together, they tend to be water phobic or water fearing, and that influences the structural shape of the compound. Now, another bond that exists beyond even that of the hydrogen bonds that we kind of highlighted here and the hydrophobic bonds that were highlighted here are things called salt bridges, which can cause interactions as well. Here you have your amino group interacting with the carboxyl group of a compound. And it's almost like what happens when you mix an acidic type amino acid, like glutamic acid or aspartic acid, with another particular compound, like arginine or a basic compound, rather, and they form these salt bridges. Now, in addition to these hydrophobic interactions, these hydrogen bonds and salt bridge interactions, one of the things you have to keep in mind is the hydrophilic interactions, which would probably be mediated or encouraged by that of your hydrogen bonds. Now, one other bond that hasn't been mentioned, and it's limited to interactions based on the presence of mainly one amino acid, which I mentioned earlier as cysteine, is that of a disulfide bond. Now, disulfide bonds are interactions where you have a thiol group here and a thiol group here, and they form a disulfide bond. And these type of interactions are heavily influential. In fact, they have a significant prominence when you talk about hair and the protein associated with hair. And so these disulfide bonds help also influence the structural makeup of a protein. So we're highlighting hydrogen bonds. We're talking about hydrophobic interactions, which are non-covalent interactions as well. You're talking about salt bridge, where you have a basic and acidic type amino acid residue interacting with one another. And then you're having a disulfide bond, which can also impact the interactions. Now, all of these contribute to the whole aspect of proteins being folded. So that's why it's important that you understand these non-covalent interactions, because they help influence the shape and integrity of a protein. Now, ionic attractions between R groups are formed used through the salt bridges, which I mentioned. You have your ionized acidic and basic side chains, and they create the salt bridges. I'm reiterating our interactions here because it's important that we know these. Hydrophobic interactions between R groups also influence the shape of a protein because they tend to try to create environments where they exclude water. Lastly, you have your covalent sulfur-sulfur disulfide bonds, which happen when you interact a cysteine amino acid residue with another cysteine amino acid residue, and that impacts the folding shape of a protein. Now, there are a myriad of ways of investigating proteins. One of the ways that they can investigate protein is through analysis through something called electrophoresis or gel electrophoresis. Protein molecules are placed in a solution, and they can be separated by actually taking advantage of their overall net charge. And this is created when you have an electric field. We have two electrodes, one being positively charged, and then on the other end negatively charged. And then the particles begin to move towards the positive electrode. And you can measure this transition. Now, by varying the nature of the buffer, the proteins can actually be separated in a variety of ways. This may also include that of molecular weight. Once the separation is complete, the proteins can be made visible by the addition of a dye, which kind of can be used as a standard. Now, electrophoresis is routinely used in clinical laboratory for determining which proteins are present and in what amounts are in kind of a blood sample. Here's an example showing kind of that transition of movement. If you look at the image here, you have your cathode and you have your anode, and then this might be your normal gel electrophoresis symbol, and then you'll compare that to that of other markers. And so if you know this is the normal transition and this is sickle cell anemia, if one conducted a gel electrophoresis, they can determine whether someone possesses the sickle cell trait using electrophoresis. So it's very ideal, very useful as a approach to analysis of proteins. And you can use that beyond just the scope of looking at sickle cell, but it is one classic example. Now, I mentioned that the primary structure of a protein just consists of the amino acids in a sequence. Now, when you look at the secondary protein structure, you start looking at some of these interactions. You have your alpha helix, and a coil is held in place by hydrogen bonds between each carbonyl oxygen and the amide hydrogen 4 amino acid residues above it. Now, the chain is a right-handed coil, and the hydrogen bonds line parallel to the vertical axis. So you start seeing some of these hydrogen interactions of your compound, and you can see that in these structures. Now, what you also get in your secondary protein structure is something called beta sheet formation. The hydrogen bonds begin to stabilize interactions between neighboring protein chains. This kind of keeps them kind of static or a little bit more held in place. And then the protein chains usually lie side by side, so the alternating chains run from the N-terminal end to the C-terminal end, and from the C-terminal end to the N-terminal end as kind of anti-parallel arrangements, back and forth. So, and then they start to stack in pleated sheets, and that's illustrated in the image to the right. So you got, first they start to get aligned with the hydrogen bonding. You have your amino terminal end going towards your carboxyl terminal end, and on this end you have an amino end on this end, your carboxyl. So they kind of flip the direction that they're going. This is, your N to C, and your C to N, and then this is your N to C, and then they alternate, and then hydrogen bonds stabilize them, and then they stack one on top of another. That is the beta pleated sheet formation. So a fibrous protein is a tough insoluble protein whose protein chains form fibers or sheets. Examples include wool, hair, fingernails, and keratin, which is composed of alpha helices that are twisted together. Now, natural silk and spider webs are made of fiber and composed of stacks of these beta sheets, and the R groups must be relatively small so that fibrin contains regions of alternating glycine and alanine. And so these just give you examples of the impact of these types of sheets and the formation of proteins. Now, globular proteins are water-soluble proteins whose chains fold into compacted shapes that feature hydrophilic groups on the outside. Their structures can widely vary, and depending on the structure, sections of your alpha helix and your beta sheet can be present. Now, the hydrophilic side chains on the outer surfaces of these globular proteins account for their water solubility, and many globular proteins act as enzymes. And so the overall shape of globular proteins represent another level of structure, tertiary structure, and that's when they start forming kind of the globular structures. Now, the way in which an entire protein chain is coiled and folded is specific to a three-dimensional shape and serves as the tertiary protein structure. So if you have your amino sequence in your primary structure and you have beta pleated folds in your secondary, your tertiary represents the combination of intense folding that's influenced by your hydrophilic regions and other interactions. Now, each protein molecule folds in a distinctive manner that is determined by its primary structure and results in its maximum stability. A protein with the shape in which it functions in living systems is known as a native protein. A protein composed only of amino acid residues is a simple protein. Now, the tertiary structure is drawn in the style that shows the combination of both an alpha helix and your beta sheets regions, the loops connecting them, and also disulfide bonds. So let's look at that structure. When you're talking about a tertiary structure, you go from just a line of amino acids to now this folded infrastructure of a compound that features your alpha helixes, which kind of spiral, and then you have your beta pleated sheets, and then you have various other connection interactions that influence the folding. And you also have your disulfide bonds, which are indicated by the images in yellow. Now, continuing forward, conjugated proteins are aided in their function by an associated non-amino acid unit. So that might mean that there's something that's fused or connected to help influence their shape or structural integrity. So the oxygen carrying portion of myoglobin has a heme group embedded within the polypeptide chain. And so that's an example of a conjugated or partnered protein. Now, when you look at the quaternary protein structure, you're looking at the conjugation or the fusing of two or more protein chains that tend to aggregate to form these large ordered structures. Polypeptides are primarily held together by these non-covalent forces that we've discussed before, but your covalent bonds and non-amino acid portions may also be included as part of the quaternary structure. So to reiterate, your primary structure is your amino acid sequence. Your secondary structure is your alpha helixes and your beta pleated sheets. Your tertiary structure refers to the initial start stages of your folding and interactions of just that individual polypeptide. And then your quaternary protein structure represents an amalgamation or connection or fusing of other protein polypeptides together to form this conjugated protein. Now, hemoglobin is a conjugated quaternary protein composed of 4 polypeptide chains. So that means you've got 4 chains that are all connected together and interacting to form this larger compound. The alpha chains have roughly 141 amino acids, and the beta chains have 146 amino acids. The heme groups each contain an iron, and hemoglobin is the oxygen carrier in red blood cells. In lungs, oxygen bonds to iron +2 so that each hemoglobin can carry a maximum of 4 oxygen molecules. And in tissues that need oxygen, in need of oxygen, O2 is released and carbon dioxide is picked up and carried back to the lungs. So it has a very vital function. Here's an example of the structure of the hemoglobin, and you notice you have the iron molecule in the center. Now this represents a simplistic drawing of it on the right, but it has more of a 3-dimensional architecture as evidenced by the image on the left. Now, there are 4 polypeptides which we indicated, and they all are indicated by different colors. You have your purple, your green, your blue, and yellow. And the heme elements are in red. So each of them resembles myoglobin in structure, and then a heme unit is present in each of those 4 polypeptides and hemoglobin. Now, another example of a quaternary protein structure is collagen, which we kind of talked about before. Collagen is the major constituent of connective tissues. The basic structure unit of collagen is about 3 intertwined chains of 1,000 amino acids. Each chain is loosely coiled in a left-handed, counterclockwise direction, and 3 of these coiled chains wrap around each other to form a stiff triple helix in which the chains are held together by hydrogen bonds. All collagens feature a glycine every 3 residues, and prolines are hydroxylated in a reaction that requires vitamin C. So, to recap, one of the big take-home messages is understanding how protein structure occurs. You have your primary structure, which represents your amino acid sequence. You have your secondary structure, which represents your alpha helices and your beta pleated sheets. Tertiary structure, which represents the folding based on those non-specific, non-covalent interactions. They are specific non-covalent interactions. And you have a number of interactions that can contribute to this: hydrophobic, disulfide bridges, salt bridges, if you will. And then you have your quaternary structure, which represents the combination of various peptide protein sequences that are fused together to form a larger structure. And then the class of proteins have already been summarized as well. You have your fibrous proteins, which are much more tough. You got your globular proteins, which are water soluble. And you have your simple proteins, which contain only amino acid residues. And then finally, you have conjugated proteins, which include 1 or more non-amino acid units. This culminates our presentation on proteins. And if you're interested in information about the impact of other protein structures, one of those examples would be that of scurvy, which is a long time old condition that we have not faced since the 1800s or 1700s, if you will. And then you have osteogenesis imperfecta, which is a primary symptom in a disease that has kind of spontaneous broken bones. And so when you talk about those conditions, those can be heavily influenced by the amino acid and the misbehavior of those proteins. And going back to the reaction of forming proteins or amino acids, remember that water is removed during those reactions, and they form dipeptides and so on and so on. Now, the last thing that I need to go ahead and mention is that of denaturation, and it's the impact of reducing or decreasing the natural activity of a protein. This can impact the structural integrity of the protein. One example I use is if you ever scrambled an egg, you change it from one condition to another, and you have effectively changed the properties of that protein. There are a number of ways of causing this to happen, and in most cases, it is irreversible. Heat is one of the primary ways that this can occur, but there are other ones out there. In addition to heat, you have mechanical agitation, you have detergents, you also have organic compounds which can compete for bonding sites. You have pH, which can affect the integrity of the compound, and you have the featuring of inorganic salts, almost like adding salt to a solution to cause those interactions. So there are various ways that the proteins can be misshapen, folded, impacted, agitated to change their overall functionality. And denaturation just describes the ability to do that in a way that takes away their ability to be reactive or functional. Prions are infectious particles, if you will, and they're types of proteins, and there's a lot of information that support their impact to a lot of degenerative conditions. There are some well-supported facts about prions, which include that humans and all animals tested thus far have a gene for making normal prion proteins that reside in the brain. And a misfolded prion can induce a normal prion to flip from normal shape to the disease-causing shape. So there's a lot of information associated with the prion protein, and this is a specific protein. It's also linked to other conditions like Alzheimer's. And so it's important that these proteins maintain healthy living, and they have a lot of information about them, and they can be linked directly to conditions that currently exists and are degenerative. And this completes our presentation on amino acids and proteins.